In this video lesson, we're going to look at how to answer the AS mock on gas exchange. The main purpose of the video lesson is to help you understand how to read questions correctly and interpret them correctly so you give accurate answers. So the first question reads, all living organisms exchange substances with their external environment. The following data applies to an average person. The surface area of the body is approximately 1.8 meters squared. The volume of the body is approximately 0.07 meters cubed, and the surface area of the lungs is approximately 70 meters squared. It then says comment on the significance of this information for gas exchange. So how is this information relevant to what we've learned about gas exchange? Well, if you remember, single-cell organisms have a very high surface area to volume ratio. For this reason, oxygen can just diffuse into the cell and carbon dioxide can diffuse out of the cell. They require no specialized gas exchange surface. This is the first lesson we had on gas exchange. The point is, the bigger you get in size, the lower your surface area to volume ratio. In other words, now you have a relatively large volume to a relatively smaller surface in comparison. This means we can't rely on gas exchange through the skin. We need a specialized exchange surface. That's what this question is testing you on. It's making you acknowledge that, look, if you look at the body, it has a very low surface area to volume ratio. However, the surface area of the lungs is huge. So it's for 3 marks, so this is how you get them. The body has a low surface area to volume ratio, so we cannot rely on gas exchange through our skin or our surface. Or you could say we require a specialized exchange surface, or you could say we require a mass transport system. The lungs, however, have a much higher surface area for gas exchange. The point is, if you have a low surface area to volume ratio, you need a specialized gas exchange surface, and you're just using this data here to prove it. You could actually go one step further and actually work out the surface area to volume ratio, but if you don't have the time, it's not necessary for the marks. Next question says it shows the changes in the volume of air in the lungs of a student at rest during one breath. Now the question is, name the measurement represented by the line X. Now I had a few answers here like forced vital capacity and things like that. Remember, this is just at rest. We're not basically breathing out very hard or breathing in very hard. We're not forcing anything out, so it's not forced vital capacity. This is simply tidal volume. So tidal volume is your natural breathing rate. So as you breathe in, the volume of air in your lungs increases. As you breathe out, it decreases. It's all natural at rest, nothing being forced. Then it says what is happening to the elastic fibers in the walls of the alveoli at point A. So at point A, what's happening is we are breathing in, so more air is entering our alveoli, so our alveoli must be getting larger in volume. So they're stretching. The walls of the alveoli are stretching. I accepted expanding. Now, in my opinion, this is one of the easiest questions in the exam paper, and we have definitely done it in class, but no one or very few people recognize what it was asking you to do. It says, explain what causes the change in the volume of air between points B and C. Explain what causes the change. Believe it or not, we teach this to year 8s, and it's the same kind of answer. So firstly, let's see what's happening. Between points B and C, the volume of air in the lungs is decreasing. So in other words, they're exhaling. So really, this question is asking you to explain what happens when we exhale. That's it. So, intercostal muscles and diaphragm relax. If you want extra credit, you can say external intercostal muscles, though you don't need to get the marks. But external intercostal muscles and diaphragm relax for 1 marking point. The rib cage moves downwards and inwards, so the volume of the rib cage or the volume of the lungs decreases. Therefore, the air pressure is greater in the lungs than the atmospheric pressure. So this is before the change and this is after the change. So you can see that the volume of the lungs is decreased and now the same amount of particles are crammed into a smaller space. So the pressure is greater. More particles are knocking against the surface, causing or creating a greater pressure. So before, we had a lower pressure in the lungs and a higher one outside, so that's why air was moving in. But now, after those changes, we now have a higher air pressure in our lungs and a lower environmental atmospheric pressure. So now we have a pressure gradient, so air will move along the pressure gradient. So pressure greater in lungs than atmospheric pressure, so air moves out along a pressure gradient. So that's what you need to say for 4 marks. It's simple, logical, step-by-step, cause and effect biology. Going into an exam, you must be absolutely secure on stuff like this because you'll get much harder questions than this. This was very easy as well, but obviously some people got very confused with this. Calculate the breathing rate of the student in breaths per minute. So first we just have to establish what one breath is on this chart. I appreciate it's a little bit misleading here where they say they've measured it during one breath. So you might interpret this incorrectly and assume this is one breath, but think logically. What is a breath? A breath is inhaling then exhaling. Okay, if you measured the whole thing, then you're saying inhaling, exhaling, and then inhaling a little bit. That doesn't make sense. So really, one breath is from this point where our lungs are as empty as they can be to the same point in the cycle, in the next cycle. Then you have to work out how long one breath took. So from 0 to 5. So they've made this one really easy, actually. Normally it can be much harder than this. They can give you harder figures to work with, but this one's nice and easy. We've just got 5 seconds. So one breath equals 5 seconds. So in other words, how many breaths in a minute? Well, if one breath is equal to 5 seconds, then we just have to divide the number of seconds in a minute by 5. So 60 divided by 5 will give you 12 breaths. Please remember to show your working out as well, because you can get marks for that even if you got the wrong answer. It then says about 1 decimeter cubed of air cannot be expelled from the lungs. This is known as residual volume. Suggest why it is not possible to expel all the air from the lungs. Well, one of the reasons is as you exhale, you'll be continually releasing air out of your lungs into the environment, and therefore the pressure in here will drop. So there'll come a point where the pressure outside your lungs and the atmospheric pressure is equal to the pressure in the lungs, and that means there'll be no pressure gradient. So the absence of a pressure gradient means that air will no longer move out of your lungs. But also because of rings of cartilage that keep your trachea open and your bronchi open partly, open, that means your lungs can't collapse completely. They can't flatten completely to let all the air out. So it's only for 2 marks. So 1 mark, it's impossible to collapse or flatten lungs completely. Or you could say trachea is made of cartilage, so lungs cannot flatten completely. And another mark for saying during expiration, air pressure in lungs will equal atmospheric air pressure. Or you could say there will be a point where there's no pressure gradient, so no more air can move out. Now it says for efficient gas exchange to occur, a steep diffusion gradient must be maintained between the air in the air sacs and the blood, so the air in the alveoli and the blood. A steep diffusion gradient can be maintained by ventilating the lungs. That's when we change the shape of our lungs to allow air in and out. This refreshes the air in the air sacs, replaces the old air with new air. Explain how refreshing the air in the air sacs helps to maintain a steep diffusion gradient. In other words, how does it help you keep an area where on one side of the gas exchange surface you have high levels of oxygen, on the other side low levels, so you get diffusion? So first, let's imagine what would happen if you didn't replace that air. So what would happen is initially you've got air coming in, and here are the little oxygen molecules, and then those molecules of oxygen would diffuse into the blood. But now we have the same concentration of oxygen here, on this side, in the blood and in our lungs. So there's no longer a diffusion gradient, so diffusion will stop. So by basically ventilating or refreshing the air in there, we have new air with a higher oxygen concentration, so the diffusion gradient is maintained. In other words, it's always higher on one side and lower on the other side. So air in the lungs will have a low oxygen concentration as it has diffused into the blood. You could say that. Or you could say refreshed air will have a higher concentration of oxygen compared to the blood as oxygen is transported to respiring tissue. So the idea is you've explained why it's high on one side and low on the other side, therefore there is a steep diffusion gradient maintained. Now it says describe and explain one other way in which a steep diffusion gradient is maintained in the lungs. So far we've only looked at the idea of refreshing air, but the other side we haven't really looked at the blood. So if this represents our blood and this is the oxygen in our blood, in red blood cells, then if you remember, blood always transports oxygen to respiring tissue so it can be used in respiration. So therefore, oxygen is continually being transported away from the lungs, so you always have a low concentration on this side. So that also helps maintain a steep diffusion gradient, other than refreshing air. So alveoli have a large network of capillaries. That's describing, so you're saying what structures are in place to help explain this. So oxygen is continually transported away from the lungs by the blood to respiring tissue. That explains it. There's the description, what it is, the structure, and or the adaptation, and there's the explanation, how that helps. So this was the one question I genuinely thought was very challenging. I thought all the others were quite easy if you'd revise for them, but this one is challenging. This shows a spirometer, which is used to investigate lung function. What I wanted you to do, what I expected you to do, is really look at this and look at what's going on and try and, like some sort of toy you might play with as a child to try and figure out how it works. I want you to have that kind of feeling of play, to just think, right, how does this work? What do you do here? What does this do? Just looking at every part and piecing it together, and then you would have got somewhere. Some of you just had an instant sort of fear response and thought, I'm leaving that one. So let's look at this. We've got a mouthpiece, so clearly someone's going to blow and suck air from this point. We have two pipes and water here, and then an air chamber in here with a lid which has a kind of anchor point here. So what you can imagine, and also you've got soda lime there. This is basically, in every spirometer there'll be something to remove carbon dioxide. Here they put soda lime. So the idea is that you would breathe in and breathe out, and that would change the amount of air in this chamber. And that's why you put it on water, because this lid is anchored on one point, so it'll move up and down in response to the volume of air inside the air chamber. So let's say first we're inspiring, we're basically taking in air. Now the first difficulty would be, okay, how do I know which tube is used for inspiration, which one's used for expiration? The trick is, where's the carbon dioxide? Well, we breathe out carbon dioxide, so if we've got an absorber in a pipe, it must be for when we're breathing out, because there's no sense in absorbing carbon dioxide if we're breathing in. So we can assume that when we basically take in a breath, it goes through this pipe, and when we breathe out, it goes through this tube here. So when we're inspiring or breathing in, you expect the volume of air to go down, and as a result, this air chamber lid will go down. And when we're breathing out, you expect the air chamber lid to increase, to rise. Some spirometers literally have a pen attached to this which will draw a picture on a graph. So it will actually construct a spirometer trace graph. The other thing to bear in mind is, if we're taking air in through this pipe, and then we're releasing air from our lungs into this pipe, but every time the carbon dioxide is being absorbed, and remember, you've got no fresh air coming in, you're hooked up to this machine, so you're just using the air supply in here, then over time, this lid will be going up and down, but overall being going down every single time because carbon dioxide is being removed from the air continually. Okay, so describe how the spirometer would be used to measure tidal volume. So remember, tidal volume is your normal natural breathing rate at rest, no forced breath or no forced vital capacity or anything like that. The first thing you do is you clip your nose and breathe into the mouthpiece with the mouth only. You don't want to have your nose open as well, because maybe some of the air would escape out your nose and not all of it will go from your lungs into this system. You then breathe regularly and continually. You could even say at rest, in a resting state. That's probably quite important to add there, actually. And that's because we're measuring tidal volume. Then you just have to acknowledge what the lid does. When you inspire, the air chamber lid moves down, and when we expire, the chamber lid moves up. That will get you all 3 marks, by the way, but you could also say you use the change in lid height to draw a graph to show tidal volume. It's not really that hard once you actually understand it. What I couldn't understand is why people left this question blank. I mean, you could just have a guess, just draw a shape. Use the axes below, complete the spirometer trace that you'd expect to see recorded from a healthy 16-year-old over 10 further breaths. So they're clearly trying to make an effort here to say this person has no health problems, no emphysema, nothing like that. Over 10 breaths. So this is 1 breath, you've got to draw 10 more. If you draw it as a straight line, you get 1 mark, but you had to acknowledge that carbon dioxide will be removed every time, so the trace will decrease like this. How the gradient of this doesn't matter, it just has to decrease. Whether you did it really steep or really gentle, it doesn't matter. So this is a bit like the locus question I showed you, and again, this is the hardest question in the paper. Describe how you could use a spirometer trace to measure the rate of oxygen uptake. Well, before we do that, remember how this system works. So we breathe in, we take the air from here, and then we breathe out, but the carbon dioxide is being absorbed. Now remember, so in other words, this is air minus the carbon dioxide we breathed out. Now where has that carbon dioxide come from? Well, it's come from the process of respiration. So remember, that carbon dioxide there has come through the process of respiration. How was it formed? Well, to basically produce that carbon dioxide, we had to react oxygen with glucose. So we can assume that the amount of oxygen we've used in respiration, so the amount of oxygen into our body, equals the amount of carbon dioxide that's produced by a body and released. What I'm getting at here is that basically, if you look at the decrease in the crest of the wave, so it's going down this distance to this distance, the difference there, that represents how much of the air has been removed from the chamber as due to it being carbon dioxide. And therefore, it's the same as the amount of oxygen being used by a body for respiration. So the first point is the difference in the crest of wave height. This is the crest of a wave, the peak of the wave. The difference in the crest of wave height between waves indicates oxygen used in respiration. However, it's not just saying how much oxygen, it's saying the rate. That means over time. Rate means over time. So you have to measure how much oxygen is being used over a given time and then simply divide the oxygen used over time. So you divide oxygen used over the time measured to work out the rate of oxygen uptake by cells for respiration. So for example, if you were measuring over 60 seconds, you'd measure the difference between this height and work it out from reading off here. So roughly from this point to this point, so there, and divide it by 60 for 60 seconds. Finally, you really shouldn't have got this wrong. I mean, these are the most basic biology questions. This could be a GCSE question, it could be a Key Stage 3 Year 7 question. I'll be honest with you, if you're not getting these right, it shows a complete lack of revision, and you have to assume that your competitors, other students out there, they will be getting these questions right. So if you get these wrong on such a fundamental level, you are kind of out of the race. You cannot afford to do this. There's not any explanation here. You just have to know that these are the bronchioles and these are the alveoli. Remember, you have the bronchi up here and then the trachea at the top. Then it says, state two features of the structures labeled B that enable efficient gaseous exchange. So B are the alveoli. Again, very, very basic biology, could easily be GCSE question. They create a large surface area, that's why there's so many of them, and they have thin walls for diffusion. So they have a thin epithelium layer or just thin walls will do. All it says is state two features, doesn't say explain them or describe them, just state what they are. Finally, explain why a single-celled organism such as Euglena does not need a specialized area to carry out gaseous exchange. Well, this goes back to the first point I made when we started this tutorial. Small, single-celled organisms have a huge surface area to volume ratio. So because they have a very high surface area to volume ratio, oxygen can diffuse across the cell membrane. 2 marks. I hope this tutorial has shed some light on how easy this actual paper can be if you can just read questions properly. You'll see a lot of this is stuff we've covered in class and we've gone through and you understood in class. Yeah, they phrase it in ways which maybe are initially a bit difficult to access, but once you get into the rhythm of it, it's really easy stuff. The stuff you have to watch out for are questions like this, where you do have to look at experiments and apply your knowledge, and it will always be difficult, it will always be out of your comfort zone, and we can't prepare you enough for this. It's just impossible. So you're just going to practice this, do as many exam questions as possible. Right now, I appreciate there's not a lot out there. You could even look at the OCR spec where some of these questions have come from to get a bit of practice in. Their exams are actually easy to download. You can download them for free just by Google searching OCR A-Level Biology exams.